Hello students, in this lecture we are going to study about the pistil, megasporangium and embryo sac. In previous lecture you have already studied about male structure in flowering plants. Here we are going to study about female structures in flowering plants. In flowering plants, mainly three structures are important here as a female reproductive structure and mechanism: pistil, megasporangium and embryo sac. First of all, gynoecium. As you know, it is a female reproductive wall in flowering plant. In gynoecium, carpels are present. Carpel is a unit of female reproductive system. In some flower, only single pistil is observed and it is known as monocarpellary gynoecium. In some flowers, more than one pistils are arranged and it is known as multicarpellary gynoecium. Multicarpellary gynoecium, two types are there: syncarpus and apocarpus. In first type, syncarpus, here carpels they are fused. In apocarpus they are free. So this way we can classify gynoecium in flower. Now pistil. We can also say pistil is a structural and functional unit of female reproductive system. It is having three parts. First, stigma, second, style and third, ovary. As you know, stigma is uppermost part of pistil. It is actually landing platform for pollen grains during pollination mechanism. Second, style. Generally, it is a elongated thread-like structure and located beneath the stigma. Third, ovary. It is basal, lowermost region of a pistil having placenta inside. At maturity, it develops megasporangia or ovules for further mechanism of reproduction. So this way, pistil is having three parts: stigma, style and ovary. Now let's study pistil through figure. In this figure you can see typical structure of pistil which possesses all three parts: stigma, style and ovary. Topmost region is stigma having more than one carpels here. Between stigma and ovary, style is there. During pollination process, pollen grains they land on stigma, pollen tube develops and through style it is elongated towards ovule region where egg cell is present. The lowermost part, that is ovary, which possesses ovules. At maturity, egg cells develop and through pollination process, pollen grain it migrates male cell towards egg cell and fertilization process take place. Another figure which is showing embryo sac structure. It is actually typical embryo sac structure. Here you can see arrangement of 8 cells. Okay. Topmost region, that is chalaza end, having 3 antipodal cells. Lower region, lower end, that is known as micropylar end. It is also having 3 cell structure. In the central region you can see 2 cell structure, that is known as polar nuclei. So this way chalaza end possesses 3 antipodal cells, micropyle end possesses 3 cells, centermost is egg cell here and two surrounding cells, they are synergid cells. In the central region, two cells are observed and they are known as central cell. Now, the megasporangium or ovule structure. Megasporangium. As the name suggests, the structure which develops megaspore, that is known as megasporangium. When we study this structure in flower, it is a small structure attached to the placenta region and stalk is funicle. Here, body fused with funicle in hilum region. What is hilum? Hilum is actually junction between ovule and funicle region. This structure also possesses integuments. Integuments means one or two protective envelopes surrounding this embryo sac structure. Micropyle region. Here, small opening where no integuments covering is there. Chalaza. It is exactly opposite to the micropyle and basal region in embryo sac structure. The main structure, nucellus. It is actually mass of cells within integuments. If we talk about the property of nucellus, it is abundant in reserve food material. It can provide nourishment to developing embryo. So nucellus is very most important structure for embryo development. So these all are regions or structure observed here. Now embryo sac. It is also known as female gametophyte. Its location in nucellus. Here ovules are present. At maturity, megaspore develops. Through megaspore, further mechanism, further process turn into embryo sac. Generally, in typical flower, it is single in number. So, from ovule to megaspore to embryo sac, this way further development take place. Megasporogenesis. It is a process or mechanism through which megaspores develop. As you know, ovules at maturity, they develop megaspore mother cell. It is generally single. It is also known as MMC in short. By further cell division, it develops megaspore. Okay. What is nature of MMC? It is a large cell having dense cytoplasm. It is having prominent nucleus. So through process of megasporogenesis, ovule develop megaspore mother cell and ultimately, finally, megaspore development take place. Now structure of female gametophyte. We can also say it's a embryo sac. Now megaspore mother cell through meiosis division, reduction division develop 4 megaspores. So from one single diploid MMC through meiosis 4 megaspores develop. These 4 megaspores, they are classified into 2 types on the basis of their function. One remain functional and rest of 3, they degenerate. Okay. From single megaspore which remain functional, further development, further cell division take place and embryo sac develop. It is known as monosporic development because only one single megaspore further develops. Now which type of cell division take place within this functional megaspore? As you know, this functional megaspore is now haploid N in nature. So, further cell division is compulsory mitosis type of cell division. Through cell division, it remains N in nature and number of cell increases. When this functional megaspore undergo mitotic cell division, two nucleate structure develops first. Second time it undergoes same type of division and four nucleate structure develop. Third time it also undergo mitotic cell division process and at the end of this process 8 nucleate structure develop. So here we can study from one single functional megaspore at the end of mechanism through 3 times cell division 8 nucleate structure develops. In flowering plants, typical female gametophyte or embryo sac structure. In figure you have already studied about it. It is showing two ends, micropyle and chalaza end, and having 8 nuclei structure. First of all, these 6 nuclei at the both end, 3 arranged. They are surrounded by cell wall. Rest of the 2 nuclei at the central region, they are known as polar nuclei. They are arranged there. So, arrangement is 6 plus 2, 8. And if we further classify it, so we can say 3 plus 2 plus 3. This way the sum total is 8. Now distribution of these cells. First, egg apparatus. Egg apparatus is having 3 cell structure at the micropyle end or region. Out of these 3 cells, the central cell is known as egg cell and 2 surrounding cells, they are known as synergid cells. The synergid cells, they are having filiform apparatus. It is a guiding pollen tube into synergid cell this purpose. So, egg apparatus is made up of 3 cells, 2 synergid cells and 1 egg cell. Another structure. Second, antipodal cells. It is also having 3 cell structure at chalazal end or region. Third, polar nuclei. It is centrally located and 2 cell structure. So, simply we can say the typical embryo sac structure is actually having 8 nucleated structure and 7 cell structure. Why 8 nucleated and 7 cell? Because at the central region 2 cells, they are fused and make a single cell like structure. That's why basically it is 8 nucleated structure and 7 cell structure, and it is observed in typical embryo sac of angiosperm. So this way typical female gametophyte or embryo sac at maturity shows 8 nucleated and 7 cell structure. Now you have idea about both male and female sexual reproductive parts organs in typical flowering plants. Now the main event, pollination. What is it? It is actually mechanism of transfer of pollen grains from the anther to the stigma of plant. This mechanism is termed as a pollination. At the end of this mechanism, pollen grains which develop in anther shed from the anther to the stigma. In flowering plants, for the same mechanism of pollination, some adaptations are observed. Here, some external agents or factors are responsible for pollen grain which migrate from anther to the stigma structure. These agents may be biotic or abiotic agents. Abiotic agents are generally water and wind, and biotic agents, they are animals and insects. In typical flowering plants, mainly three types of pollination take place: autogamy, catogamy and xenogamy. So this way, three types of pollinations are there. At the end of pollination process, pollen grains which develop in anther, it migrates towards stigma of female structure. Here, these pollen grains develop pollen tube. Through this pollen tube, male cells present within this pollen tube travel towards ovule of female structure. Here, egg cells are present. Male cell fuse with egg cell. At the end of fertilization mechanism, by fusion of egg cell and male cell, zygote is formed. As you know, male cell is N in nature, egg cell is N in nature, and they fuse and form zygote, which is diploid or 2N in nature. So this way we can study in detail about type of pollination. Now some questions for practice here. First, with a neat labeled diagram, describe the parts of typical angiosperm ovule. Second, explain monosporic development of female gametophyte. Third, explain 7-celled 8-nucleate nature of female gametophyte with figure.